MCAT 2017 Cram, Psychological and Biological Foundations of Behavior, Passage 1, Perky Effect. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. There are 5 questions and they're really not that bad. So good luck and happy reading and happy thinking. Paragraph 1. The Perky Effect describes the relationship between real visual information, perception, and mental imagery. Discovered by C.W. Perky in 1910, her experiments were able to show that visualization of images can depress the sensitivity of perception of real visual targets. In her study, participants were asked to imagine certain objects on a white screen, such as a fruit, a tree, or a book. After a few trials, a faint real image was projected on the screen of the subject matter. Participants were quite often unable to distinguish between their imagined projections and the true images. For example, one participant was surprised that the banana appeared to be in a different orientation than the one they were attempting to imagine it in. Another maintained that the title of the book they were imagining was legible, while there was no title in the actual projected image. The Perky effect has since been updated to describe the reduction in visual performance, like here, see the gradual reduction, due to the maintenance of a mental imagery. Okay? So here, this is the Perky effect on like 4 different people visualizing 4 different projections of some true face structure. Okay? All right. Paragraph 2. A research group was interested in both replicating Perky's findings and in investigating the influence of contrast on this effect. Before the experiment, researchers asked some participants to imagine lines on a whiteboard, these lines here, and choose a color chip, these dots adjacent to the lines, from a prepared set between black and light gray that matched most closely with the color of their imagined lines. These lines were called fixation lines. Other participants were given a specific color of either black or gray fixation lines displayed on the white target, while the control group had no fixation lines. The imagined real color of the fixation line was referred to as the participant's map. Paragraph 3. During the task, each participant received a picture showing an image of 2 black circles, these circles here. The picture was removed from view and a target with 2 black lines was shown, these lines here. One originating from each previous fixation point, okay, with a 1/2 inch gap in between the lines. Figure 1. An image displaying both the black circles and black lines on, close, or far from the imaginary real lines. Okay? These fixation lines are shown as they appear in the gray condition as dashed lines in the figure. So this is the dashed or gray lines is where the the subject has fixated their real/imagined images that they projected in their mind, their mind's eye. Okay. For the imagined condition, the participant was asked to imagine the previous image on the two fixation lines. The nearest line was 0.1 centimeters, close to the fixation lines, with the nearest line being 0.8 centimeters, or far from the fixation lines, with the nearest line being 2 centimeters. For the black and gray line conditions, for the black and gray line conditions, the 4 fixation lines were shown continuously, and the black circles were removed and replaced with the black lines. Okay, so first the black circles are shown, and then they're removed and replaced with the black lines. The task was to decide whether fixation lines were offset either to the left or to the right. The results are shown in this graph in figure 2. Okay. So figure 2, it may be a little hard to see this. It's displaying the mean, the average percent of correct responses for each condition. So, as we look, seems to be there's like a higher overall percentage of correctness for far lines. And this is overall participants was calculated, and the duration of each trial is going to be between 3 milliseconds and 25 milliseconds. The baseline condition, none, was practiced until the participant was at least 90% accurate. Okay, so this is just to ascertain that they're reproducing the same level of accuracy. The effect of distance, line type, and the interaction between distance and line type were all significant at a p-value of less than 0.001. And some of you may be wondering, like, what's this p-value all about? So let's just do a quick aside on statistics. So a p-value is used when you're performing a hypothesis test in statistics. It's also referred to as calculated probability, and it helps you to determine the significance of your results. Okay? Hypothesis tests are used to test the validity of a claim that's made about a population of results. The claim that's on trial, in essence, is called the null hypothesis. Okay? The alternative hypothesis. So let me just show you how the null hypothesis is usually denoted. It's usually denoted as H, O, and then the alternative hypothesis is the one you would believe if the null hypothesis were concluded to be true. So you always have two choices: a null hypothesis and an alternative hypothesis, usually denoted H sub A. Okay. The evidence in the trial is your data. So here goes our data here. And the statistics that go along with it. Okay, so these are the stats. They're, the tabulated percentages. All the hypothesis tests ultimately use a p-value to weigh the strength of the evidence that's being presented, like here in the graph. So what's the data telling you about the population? The p-value is always going to be a number between 0 and 1, and it's interpreted in the following way. So a small p-value, typically less than or equal to 0.05. Sorry for the delay. Indicates strong evidence against the null hypothesis, okay? So in that case, you would reject the null hypothesis. And here we have really strong evidence against the null hypothesis because our p-value is less than 0.001. It's almost essentially 0. Okay, I know this still might be like really confusing to you, so let's break it down even more. Let's say, for example, a pizza place claims their delivery time is 30 minutes or less on average, but you think it's more than that. You would conduct a hypothesis test, and the null hypothesis, HO, would be that the mean delivery time is 30 minutes max. Okay, this is what you're testing, and then your alternative hypothesis would be HA, and that's that the time is greater than 30 minutes. Okay, this is what you believe, and HO is what the pizza place is claiming. You randomly sample some delivery times and run the data through a hypothesis test, and your p-value turns out to be 0.001, okay, which is less than 0.05. So in real terms, there's a probability of 0.001 that you will mistakenly reject the pizza place's claim that their delivery time is less than 30 minutes. So that's a really small probability, okay? And let's see. So since we're typically willing to reject the null hypothesis when the probability is less than 0.05, you can conclude that the pizza place is wrong and their delivery time is in fact more than 30 minutes on average, okay? And you know that, and you want to know that what they're going to do about it, of course. But you could be wrong, and you could have sampled like a really bad sample. So, okay, basically what I'm trying to say is in terms of the effect of distance, line type, and interaction between distance and line type, we're saying that, you know, they have some significance. So I don't know what their null hypothesis is, but just know that these interactions are legit, okay? You can go review something else on this if you want more information. All right, so for our first question, which conclusion can be correctly drawn regarding the results displayed in Figure 1? Is it A, no Perky effect is shown in the data? Is it B, it's not possible to infer whether the data show a Perky effect? Is it C, a clear Perky effect was shown with an increasing effect when the masking lines were visualized closer in proximity to the fixation lines? I'll give you a moment to think, and definitely press pause if you need to. You may want to open up a second window to view the passage or visuals in order to select the correct answer choice. All right, so let's break this down, okay? Let's see. The effects of line type and proximity to fixation lines were significant, you know, as mentioned in the caption for Figure 2. The Perky effect describes a significant reduction in acuity in imagined lines and black line conditions. This is something that you would have to know probably outside of the passage. So this is standalone knowledge that you would have to bring with you to the test, okay? The Perky effect was shown to increase with proximity for the imagined masking lines, so and the black masking lines. So what I'm trying to say is that a clear Perky effect was shown with an increasing effect when the masking lines were visualized closer in proximity to the fixation lines. And this is not a clear visual, so you may want to get your own separate visual printed out and have it handy with you as you're going through these questions. All right, question 2. What part of the visual system is the Perky effect likely taking place? So this sounds funny, but yeah, where is this taking place? The Perky effect does not happen in the visual system. It's a fixation problem. Option 1, and answer choice B, in the brain, C, in the fovea of the eye, or D, in the retina of the eye. I'll give you a moment to think. All right, so let's see. A problem of fixation would make it difficult for participants to complete the control non-masking condition accurately. But in the caption from figure 2, we read that in this condition, 90 participants average over 90% accuracy. That's the purpose of showing that figure. So the Perky effect, this is standalone knowledge you would have to know, is a perceptual phenomenon. However, the eye is not transmitting imagined images to the brain. So it's most likely that the Perky effect takes place in the brain itself, okay? It's not a matter of your eyeball seeing things that are not there. You're just kind of coming up with it and imagining it yourself without actually seeing it, okay? This passage is probably one of the more difficult ones because this is like a hard concept to grasp, but just stick with it and rewatch this a few times. All right, third question. What is a masking stimulus? Is it A, a masking stimulus is shown simultaneous to another stimulus which causes a failure to perceive the first stimulus? Is it B, a masking stimulus is shown immediately after a first visual stimulus, which causes a failure to perceive the first stimulus? Or is it C, a masking stimulus is shown immediately before a first visual stimulus, which causes an enhancement in the perception of the first stimulus? So definitely press pause if you need to. Okay, so a masking stimulus basically causes a failure in the first stimulus. This is outside knowledge that you have to bring to the test. Interference basically happens when two stimuli are perceived simultaneously, as indicated here. So this is interference. And so the correct answer choice is B. A masking stimulus is shown immediately after the first visual stimulus. So let's say this is the first visual stimulus in this trial and this for that, okay? If you show, I don't know, let's say this is what you want the person to see, but you show this right after, and this is what they come up with. Or let's say the bottom left square is what you want the person to see, but you show this top one right after, and this is what they come up with. in their mind's eye. So this is what it means to cause masking. And a masking stimulus, again, is shown immediately after first visual stimulus, which causes a failure to perceive the first. Okay, you perceive this instead of this. All right. So I think this is like the fourth question. Another researcher shows that the Perky effect lasts for as many as 6 seconds. How might this finding affect the results shown in Figure 1? Is it A, the new findings contradict the results shown in Figure 1? Is it B, the new findings strengthen the results described in Figure 1? Is it C, the impact of the Perky effect will be decreased with a longer trial? Or is it D, the new findings will not affect the findings of the study described in Figure 1? Definitely press pause if you need time to think. All right, this question is really difficult. It might seem easy, but it's not that easy. You really have to think beyond what's presented to you. Okay, so the caption for Figure 2 states that the duration of the trials is between 3 milliseconds and 25 milliseconds. The trial times are basically determined by the individual idiosyncrasies of each participant. So it's not really possible to infer that the results are different from those described in Figure 1, other than both support the existence of a Perky effect. Okay, so the correct answer here would be the new findings will not affect the findings in the study described in Figure 1. It's just like a different person or a different set of people, different idiosyncrasies, okay? All right, so for the final question, the fifth question, what Gestalt principle or Gestalt, I'm not really sure how to pronounce this. If you know, please let me know, describes the integration of perception and mental imagery manifested in the individual who described the book they were imaging. Is A, the law of pragnance, like all these little tiny things together, coming together in a figure that you recognize as something else? Is it the law of proximity? Is it C, the law of similarity? Or is it D, the law of continuity? So definitely press pause if you need time to think about this one. This is a standalone question. You don't need the passage to answer this. Okay, so let's break this down. The law of similarity claims that elements that are similar tend to be grouped together, by your visual faculty. The law of continuity describes the ability to group units into lines following the smoothest path. So we would group like this instead of something like this, which has more jagged edges. And the law of proximity claims that elements viewed as groups if those groups are close together. So this is grouped rather than, we don't group it like that or like this. Okay. They're close together, so we have 4 separate groups here based on their distance, the distance of the dots relative to one another, okay? But the law of pragnance, which is the correct answer, describes the principle that elements are grouped together if they are seen to be a part of a pattern. This requires more complex cognitive processes, and So the pattern has to be coherent and unified. And here we're seeing an Abraham Lincoln. I think this is a painting by Salvador Dali. He's a really good painter. And imagine title appearing on a book follows such a pattern. Okay? So final question, final answer.